So I'm going to talk to you about development of COVID-19 vaccines at the NIH Vaccine Research Center, the VRC, which is where I work.
Recording in progress.
And our recording is in progress, so if anybody doesn't like that, please log off now. I need to, there we go. So in my personal opinion, one of the greatest scientific achievements of the past century really has been the development of vaccines for the COVID pandemic within a year of the discovery of the virus that caused it. I mean, this is just unprecedented and has saved millions of lives. It's the fastest that a vaccine has ever been developed. Now, this was an amazing achievement. It also gave some people pause. I think people were worried to take these vaccines because they were made so quickly. And so a question I get is, well, how is that possible? When it usually takes decades to make vaccines, how could you have done it in a year? And how could these vaccines be any good if you made them in a year? And so how did we do that? And the truth is, it was not just a year, because it built on decades of prior research on how to make vaccines, on how coronaviruses work, and many steps that are usually done one at a time with big gaps in between them were done right after the other or done in parallel, done without any pauses in between them. And everybody, scientists, doctors, regulatory people, biochemists, engineers, everybody was working extra time to get this done. And that's how we were able to do it in a year. So I'm going to tell you the stories about the decades of prior research and the research that was done over that year to make these vaccines happen. And the story that I'm going to tell is an example of one of these vaccines. I'm going to tell you about the NIH and the Vaccine Research Center involvement with the Moderna vaccine, mRNA 1273. So I'm going to talk about the work that was done beforehand, how we got to the starting line in this, the initial testing that I was involved with, and then since the vaccines have become available, the work that we've done understanding the durability of the vaccines, how long do the immune responses last, and how well do the vaccines do against all the different variant viruses that have come around since the beginning. So one thing I want to point out is that pandemics keep coming. There have been multiple pandemics over the past 100 years. You guys have probably heard about the 1918 influenza pandemic that killed more people than World War I, which it came at the end of. There have been other influenza pandemics. The HIV pandemic continues to kill 1 million people every year. And there have been other viruses like Ebola and SARS and Zika that have swept around the world. So pandemics come. And there will probably be another one after COVID in 10 or 20 years that maybe we'll be ready for, or maybe not. And so several years ago, several of my colleagues talked about what do we do to prepare for the next pandemic, because we know there will be one, even if we don't know what it's going to be. And so they proposed a concept that they called prototype pathogens for pandemic preparedness. Say that 5 times fast. But the idea is this: we don't know what virus is going to cause the next pandemic. And there are hundreds of viruses in people, and there are hundreds of viruses in animals that we know about and plenty that we don't know about. But it's not an infinite problem. If you look at the viruses that infect humans, there's actually just 20 families of viruses. So there's coronaviruses and pox viruses and influenza viruses and adenoviruses. We know what those families are. So the prototype pathogen concept is to take one virus from each of those families, call that your prototype virus, and learn how to make a vaccine for that. So then when the next virus emerges out of the jungle, out of a bat, whatever, when the next scary virus emerges, we'll already know something about that virus's relative. We'll know how to make a vaccine for a member of that virus family, and then we can apply what we have learned to the new pandemic virus, and hopefully even before it becomes a pandemic. So that's the pandemic preparedness concept. And at the end of 2019, the NIH and the Vaccine Research Center had a partnership with what was then a small biotech company called Moderna to test an mRNA vaccine for Nipah virus as a demonstration of this approach. But in January of 2020, we thought, well, maybe we should do coronavirus instead. There's this coronavirus in China, and maybe we should do that as a demonstration project. And if it doesn't turn out to be anything, then at least we'll have done our demonstration project. But if this turns out to be the real deal, then we'll be ahead in trying to get a vaccine. And so it turned out that was for sure the right call. And so we were able to do that. We were able to switch to do a vaccine for coronavirus because there was decades of prior research that set us up to make this vaccine quickly. And so there's research in a lot of areas. I'll talk about just 2 of them. From 1989 to 2020, there was a lot of work being done making RNA vaccines practical. And so there's been a lot of work on so-called genetic vaccines, but in particular on RNA vaccines, Caroline Carrico and Drew Weissman at UPenn, and then some biotech companies, Moderna, and then one called BioNTech, which you know better as being working with Pfizer. And then there was a lot of work on coronaviruses from labs around the world, but particularly work in the last decade from researchers at the Vaccine Research Center at NIH. So just a word about RNA vaccines. RNA is it's a molecule of the genome. It's a molecule that carries genetic material, and what it does is it's the blueprint for a protein. And a protein is what the immune system likes to recognize. So you make an RNA that encodes a gene from the virus, just one gene, not the whole virus genome, but just one protein, so that it won't actually cause a virus infection because it's just one piece. So you take RNA that encodes the gene for your one gene from your virus. You encase it in a lipid coat, shown as this little ball here. So it's called a lipid nanoparticle, and this protects it's basically a little bubble of fat, and this protects the RNA when it gets injected into the body. And so you do that, you inject it into the body, and cells at the site of injection pick up the RNA, make the protein that the RNA is the blueprint for, and show that to the immune system. And then the immune system recognizes this as foreign, recognizes this as dangerous, makes an immune response against that protein, and then there's something called immune memory, where the cells of the immune system are ready the next time they encounter this virus. And that's how vaccines work. And so that's basically how an mRNA vaccine worked, and it took decades to get this to actually go. But it was work by the scientists that I mentioned before and these companies in addition. There's also a lot of research on coronaviruses, and COVID is caused by a coronavirus. And so I'm going to talk about research done here at the Vaccine Research Center, led by Barney Graham and Kizzmekia Corbett, and also our colleagues at the University of Texas. So Jason McClellan, who actually was a postdoctoral fellow here at the Vaccine Research Center, and then his graduate students, first at Dartmouth and now in Texas, Daniel Rapp and Yuan-Shuang Wang. So as I mentioned, COVID is caused by SARS coronavirus 2. And you may have seen this picture of it. So it looks like a little ball with these red spikes sticking out. And the virus uses these spikes to stick to its target cells in your nose and your lungs, et cetera. And a lot of work has been done on the structure of this protein. So this is a cryo-electron microscopy reconstruction by Jason McClellan's lab of the spike protein of the SARS coronavirus protein. So what we know about coronavirus spikes, like the spike proteins of many viruses, it's what we call metastable. So it exists in two different forms, that what we call the pre-fusion and the post-fusion form. And you can see that there's a large conformational change between them. You get this huge change in their shape. So it's the same protein, but it can have this compact form or this long extended form. And so what this looks like when you take an electron micrograph of individual spike proteins, this is negative stain electron microscopy, you can see that some of them are in this compact pre-fusion form and some of them are in this post-fusion form, just like in this cartoon that I showed you. So what does that have to do with anything? Well, it turns out that in other viruses, such as the respiratory syncytial virus, the pre-fusion spikes make much better vaccines. They give you much better antibodies than the post-fusion form. So you definitely want your pre-fusion form as a component of your vaccine. And just from a practical matter, the pre-fusion spikes are much easier to produce. You get 50 times as much when you make them in the lab. And so Yuan-Shuang Wang and Jason McClellan worked to put in mutations in the spike protein that would stabilize the protein. And so for all you biochemists out there, the protein has these two long alpha helices shown in yellow and in orange here. And when it flips from the pre-fusion to the post-fusion form, this orange helix flips up and forms one very long helix. So instead of having sort of two sticks folded back like a pair of nunchucks, you've got one very long stick. And that's how you get this extended form of the protein. And so what Yuan-Shuang figured out is that if you put two mutations right here at the junction between the two helixes, if you change these two to prolines, then you no longer can have that flip. It no longer shoots up like a harpoon and makes that one long stick. So that stabilizes this. They're right here in this structure, and that stabilizes it, makes it always be in the pre-fusion form. So when they tried this on different coronaviruses, it worked. So this is an example for the MERS coronaviruses. That's the Middle Eastern Respiratory Syndrome virus, which you can actually catch from camels. There's an ongoing outbreak of that in Saudi Arabia, and it can be very deadly in people. And so the first thing they they tried it in several coronaviruses, and in this example, it's the MERS coronavirus. So if you make the wild type, the original protein, you get a mix of the pre and the post fusion forms. But if you put these two prolines in the 2P form, now everything comes in this nice compact pre-fusion form that you want. They tried it in other coronaviruses. So here's the SARS coronavirus, so SARS-1, which caused a huge outbreak in, I think, 2003 in Hong Kong and Canada. So again, the wild type is a mix, and then the S2P is always this nice compact form that you want. And then here's the really important part, is it was tested in animals. So Barney Graham's lab tested this in mice to see if these would work as vaccines. And here's the original version shown in blue, and the 2P version, and the y-axis is showing the titer of antibodies. So basically the mice are making much better antibodies if you give them the 2P form than if you give them the original form. So based on this, we knew that 2P was going to be the way to go for a coronavirus vaccine. And others agreed. So in our partnership with Moderna, that was the version of the protein that was used. Other companies were also aware of this research because it had been published a few years prior. And so Pfizer uses the same thing. So does Janssen, also called J&J, Novavax. So basically, if you got your vaccine in the United States, you got something that had 2P in it. There are other vaccines that don't use it, but all the ones that are used in the USA actually do use this 2P form. So with all of this in hand, of the previous work on coronaviruses, with the work on mRNA vaccines, we were able to go at, quote, warp speed. That was actually, Operation Warp Speed is actually the original name of the White House task force and a large group of people working on this. So on this rather complicated timeline, I'll just show you that, up until 2019, there was all the work on the RNA vaccines, the work on coronaviruses. And then at the end of 2019, word finally started to get out from China that there was this horrible epidemic of a respiratory disease. And on January 10th, virologists in China posted the complete genome sequence of this new virus. And they posted on a database where anybody in the world could see it, which was a technical and scientific tour de force, and also I think probably politically rather brave of them. So it was put on this public database. We were able to look at it. And just a few days later, the VRC and Moderna decided what sequence they wanted to use to put into a test vaccine for this, what we hoped then would be just a demonstration project. So after that, two things were done that were really important. First of all, it was tested. We tested it in mice and showed that it indeed gave good antibodies in mice. That was important. And Jason McClellan's lab showed the molecular structure, showing that yes, indeed, by putting these mutations in, it worked just fine in this virus, just like it had worked in MERS and SARS. And so, with all of this work being done, in the middle of March, the first in-human trial started. The very first person got an injection of this. So just 65 days from first having the publication of the sequence to the start of the human clinical trial, which is a new world record. At the same time, we had to develop assays. We had to develop lab tests to see if the vaccine was actually doing what we thought it was going to do. We had to develop this because they didn't exist, because this virus didn't exist before a few months earlier. So that work had to be done, which is not quite as glamorous, but is really important and enabled things that came after. So I'm going to spend a little bit of time on that. So there are several branches of the immune system. The immune system has things called T cells, including killer T cells, and the immune system makes antibodies. Antibodies being proteins that will attack a virus or other pathogen and clear it from the body. But antibodies do a variety of different things, and so you have to know what antibody functions you want to measure. So you can measure just binding. Does your antibody, which I'm showing is this Y-shaped thing, does it stick to the protein that it's supposed to stick to? Does it have a biochemical function? So there's an assay that we do to look for ACE2 inhibition. So ACE2 is the protein on target cells that the virus sticks to. So if you can interfere with the interaction between the spike protein and ACE2, then you're probably going to be able to block the virus. So that was an important biochemical function that you can test. And then my job has been to look at neutralization, and neutralization is testing whether the virus can prevent infection of target cells. So we had to develop a neutralization assay for COVID, for SARS-CoV-2, but we didn't have to exactly go from scratch. The assay that we worked up was based on one that was originally developed for MERS. We were able to take out the MERS specific component and put in a SARS-CoV-2 component to make our COVID assay. And that assay itself was derived from assays that had been made before for influenza. So we're building on all kinds of work that was done before. In addition, the initial optimization of this assay, so we had a lab version of this assay that we could use on like a few mice, but we needed to make this good enough, robust enough, reproducible enough, and high enough throughput that we could actually use it for human clinical trial samples. And so my lab for over 15 years has been doing standardized assays for HIV. And so we applied what we knew about these assays, the assay conditions, how do you set up your Petri dishes, and the analysis software that we had and tracking software that we had and databases. And so we applied that whole structure to this assay. And then we also got good target cells from a collaborator. I always have to thank Waimu at Scripps for this. And then after all of that, I had to learn how to do a whole lot of paperwork and do the documentation to be able to call this quote fit for purpose so that we would be allowed to do this for clinical trial samples so that the FDA would actually accept our data. And so all of this took the combined expertise from other viruses and assays that we already had. And so I want to just quickly thank all the people who are doing this. I won't list everybody, but especially Kazmikyo Corbett and Lingshu Wang, who developed the original assay, C.G. Odell and Steve Schmidt, who helped me do the standardization and whose data I'm going to be showing you now. So how does this assay work? My lab doesn't use live viruses. There are other laboratories that do use the live coronavirus, but it takes a lot of work to make virus stocks that are standardized, and you have to have a biohazard level 3 laboratory to do that work. My group doesn't have that. So for that and some other logistical reasons, it's easier to do what we call the pseudotyped virus, where the virus is made out of basically broken into pieces, and it can infect cells, but then it doesn't continue the infection. It does one round of infection and it stops. So this makes it safer. And it also improves the assay in some ways. At any rate, so what you do is you have 4 components. You've got you've got the gene for the spike protein. So we have the full-length spike protein. You also have the genes for a lentivirus backbone. This is actually a broken HIV genome that's going to carry the SARS-CoV-2 spike. We also put in a protein called TMPRSS2, which is an entry cofactor. And then my personal favorite, firefly luciferase. This is the enzyme from fireflies that makes them light up. And so we can use this to track whether cells are infected or not. So you take these 4 genes on plasmids, you co-transfect them into 293 T cells, and you get out what we call the spike pseudotyped virus or pseudovirions. So then you take these pseudoviruses and you put them on target cells that are expressing ACE2, which is the receptor, the protein that the virus uses to enter cells. You let it sit for 72 hours, and then you measure luciferase activity. So you add a chemical also from fireflies, and if the cells are infected, they will light up, and then you can measure the light emission. Now, so you do that in some of your samples. In other samples, you add your antibodies that you hope are neutralizing antibodies. So this can be monoclonal antibodies. This can be your serum samples from the blood of an infected or a vaccinated person. So you pre-mix that with your virus. You put that on your target cells. And then if you have neutralizing antibodies that work, then you don't get infection, you don't get light. And so you look at the reduction in light, which tells you the reduction in infection. And that's how we measure neutralizing antibodies in the lab. So all of that, developing RNA vaccines, learning about coronavirus spike structures, developing the assays, testing this in mice, that's all what we call preclinical research. And that can take decades. The next part of this is clinical trials in people. So we have that's broken into 3 phases. So phase 1 clinical trials is your first in human trials. It's small groups of people, 10 or 15 people per group, where you look at, is this vaccine safe? What dose is safe? What doses are going to give you some immunogenicity, right? Some doses might be low enough to be safe, but don't actually give you any antibodies. So a little bit of dose finding. Phase 2 is the same thing, but with maybe a few hundred people. And then phase 3 is your trials that tell you, does this actually work? This is what we call efficacy trials. And that's where you'll have like 30,000 subjects. But everything starts with one, long journey starts with one step, right? So the first thing you have to do is phase 1 clinical trial, the small groups and some immunogenicity. I'm going to show you data now from my lab, looking at the very first people who got Moderna's vaccine. So this is a complicated graph and I'll walk through it slowly. So on the y-axis, these are titers of neutralizing antibodies. So higher numbers are better. And then each set of dots is the data for one group of people who got vaccinated over time. So people got 25 micrograms or 100 micrograms or 250 microgram injections at days 1 or 29, indicated by these syringes. And then we, in this part of the study, we followed them for the first 2 months of the experiment. And so each of these dots is the level of neutralization from each of these people at different time points. And so we noticed certain things right away. So first of all the subjects, no matter what dose we gave them, developed neutralizing antibodies after 2 doses. This was a huge relief because this is a brand new vaccine. We did not know what this was going to do in people. We knew what it did in mice. We did not know what it was going to do in people. And to our great joy, we saw that it actually gave us neutralizing antibodies after 2 doses. And then we were able to look at the different dose amounts, right? So here are the people who got 25 micrograms, people on the left and people on the right getting 100 micrograms. And we can see that 100 micrograms was quite a bit better than 25 micrograms. The levels were higher. But if we compare that to the people who got 250 micrograms, it actually wasn't any better than 100 micrograms. So that tells you that 100 micrograms is enough, which is great. It's going to be a lot less expensive if you can make 100 micrograms versus 250, and you can make more doses, right? You get this more doses out of the same batch that you make. Furthermore, the people who got 250 micrograms had more side effects than the people who got 100 micrograms. And so if you've had a Moderna or a Pfizer vaccine, you know that there are side effects to these vaccines that last for a day or two. Headaches, nausea, fever, feeling tired. And all of those things were more frequent and a little bit worse for the people in the 250 microgram group. And so based on that, these data and the data about the side effects directly informed the selection of 100 micrograms as the dose for the phase 3 trial and subsequently for the authorized dose. So that dose that a lot of us got, that's based on these data right here. We also went on to look at people in different age groups. So, and for a longer period of time. So what I showed you before was just the first 2 months. We followed them for 7 months total, for at least on this graph. And so we had 3 age groups. So we started with adults aged 18 to 55. We also have older adults, 56 to 70, and then 71 and up. And these people were particularly important because, as you know, COVID is much more dangerous in older people. So it was really important to have a vaccine that was going to work well in our older individuals. And so what we saw is that these graphs all look pretty similar. So this is binding, so just virus sticking to antibodies sticking to the spike protein. And here's the neutralization data from my lab and then from a colleague's lab. And so what you see is not much going on after 1 dose, but after, and each of these lines, a friend of mine calls these spaghetti plots. Each of these lines represents 1 person in the group followed over time. And so what you can see is for these people, very little after the first dose, but after the second dose, nice high antibodies, and then it declines slowly over the course of 6 months. But most people still had measurable antibodies in this assay, even at the 6-month time point. In the other assay, everybody still had it at the 6-month time point, even the older people. So that looked really, really good. So all of these things happened in the winter and spring. And then in July, the phase 3 clinical trial started. So that's the efficacy trial to see if it actually worked. 30,000 people were tested, and the answer was it worked really well. It worked spectacularly well. And so these are the data from that trial, and each of these tick marks represents 1 person getting infected over 120 days. And you can see that there are really very few cases in the people who got vaccinated, but the people who got the placebo, people who got a fake vaccine as a comparison, got infected. And so when you do the math, this comes out to 93% efficacy. And everybody was really happy. I mean, we would have been happy with 50%. And to see 93% just sort of really made everybody's day. And so that data and our data and others, data from other studies, when it was submitted to the FDA. And so this is a screenshot of our data being presented to the FDA by Moderna. And the Moderna vaccine was authorized, the Pfizer vaccine was authorized. And so by December of 2020, we were feeling really good about this thing. We're like, yes, we've got a vaccine, it works, we're going to be able to vaccinate people. It may take a little time getting started, but we're going to vaccinate people, we'll be out of this pandemic, and kids can go back to school, and restaurants can reopen, and I can go back to my day job, and this is going to be great. Except viruses mutate. This is what they do. Viruses mutate, they accumulate mutations in their genomes. And so we started with just 1 virus, but then we ended up with variants, with names like, well, we had lots of names initially and then kind of settled on alpha and beta and gamma and delta and whatnot. And so these variants, they're more contagious. So from an evolutionary perspective, they're better adapted to their new human host. You know, they'd only been in humans for a year. They're mutating and adapting to their new human host. And some of the mutations give you immune escape. And so they're able to withstand the attack of the immune system by mutating. And these every 6 months or so now, another variant sweeps the globe. So this is showing you the different sequences that are in the sequence database. And there have been millions of sequences that have been deposited by researchers into a public database, which is really very helpful. And so in the beginning of the pandemic, we had the original or the ancestral sequence that was quickly replaced by a genome with a single amino acid change that didn't affect its recognition by antibodies, but it did make it more contagious. And then that was followed by alpha, which was then replaced by delta. And in the middle, there were a bunch of other little things called beta and gamma, and they were in different parts of the world. Beta was in South Africa, gamma was in South America, iota was in New York, et cetera. But in general, one of them tends to win out after a while. And what does this actually mean? So if you look at the spike protein, there are about 10 or 12 amino acid changes in the spike protein. And so what I'm showing across this line is all the different positions in the protein. It's about 1,200 amino acids long. These are all the places where mutations happen. And some of them are unique to the different variants, but some of them pop up again and again. So here's that D614G. Here's a mutation that makes the virus replicate better. And here's one that gives you immune escape. So this one, E484K, we sometimes call it EEK. This mutation allows the virus to avoid being recognized by certain antibodies. This particular one makes it avoid being recognized by bamlanivimab, which was the first monoclonal antibody treatment from Eli Lilly. So this seemed like it was going to be really bad news. And so we knew that there were these mutations that were probably causing immune escape. We also knew that over the course of 6 months, you did see that drop in antibody titers. And so I organized a group of labs to look at serum from clinical trial samples over 7 months from the start of vaccination in a couple of different ways. So we did our pseudovirus neutralization assay. We also had a colleague doing a live virus neutralization assay. And then we looked at binding. So again, just does the antibody just stick to the spike protein? And we did that in 3 different formats: a cell surface protein, soluble, and 2 different soluble protein formats just on a plate. And then the receptor blocking assay. And so here's what we found. We looked at the same samples against a whole bunch of different variants. So here's our original variants and then alpha. Here's iota, the New York variant. I put it in Metz colors, and delta. And for all of them, so the doses are here at days 1 and 29. After the first dose, we've got just a little activity. After the second dose, everybody is neutralizing everything pretty well, and you get this slow decline. And the worst one is beta. This was the one that arose in South Africa. Beta was the worst. And so if you compare these data, which I now have up in this corner, compared to all the other assays, the patterns are the same. So in every case, you get a peak after the second dose that comes down slowly. And in every case, beta is the worst one. So it was rather gratifying that it told us we were on the right track, because you don't know which of these antibodies is the best representation of what's happening in a human body. So by seeing the same answer in multiple different assays at the same time, tells you that you really do kind of understand what's going on. So another way to look at this is just how many, and something I got asked a lot is, well, how many people still have recognition of these different variants 6 months after the second vaccination? So what I'm showing in this table is just how many people out of 24 still had antibodies above the level of detection against the different variants. So at day 43, which was the peak time point after the second dose, everybody neutralized everything, which was great. That told you that even though the antibodies might decline, there was still going to be immune memory in there that maybe could be boosted later. But then by 6 months out, everything had dropped. And if you look at neutralization of beta, half of people had completely lost their recognition of beta. So that was not good. And others were seeing data like this. And also we were starting to see that, that the efficacy was waning. The efficacy looks great in the first few months after vaccination, but over time, you're starting to see more and more cases. And so these data and similar data and the waning efficacy data led to the recommendation in the summer of 2021 for a third dose of messenger RNA, especially for people who were immunocompromised or older. So these are booster doses. And so I just have to, you can't have a talk on this without Dr. Fauci. So this was a White House press conference where they were talking about why they were going to be recommending boosters. And so I was really, this is kind of a career highlight for me. It was, oh, Dr. Fauci showing my graph. So that was, so that was the end of the summer in 2021, right? So summer of 2021, we thought it was going to be hot vac summer, but then Delta came in and Delta was super contagious and cases went back up and then things calmed down again. So by late fall, it looked like things were calming down. People were starting to get their boosters. We thought things were going to be okay. So it was Thanksgiving time, and a bunch of us thought it was probably okay to take a few days off. You know, I planned to take a whole 5 days off and not come into lab and not check my emails. Wednesday, I cooked, and Thursday of Thanksgiving, we had a nice family dinner. And I saw that I was getting a lot of emails, and I saw something about a new variant. I'm like, it can wait until Monday, right? I can make it till Monday. So Friday afternoon, I'm going on this nice hike with my family. Here's my husband and my kids. And we're on this night hike, and my kid turns to me and says, "So, Mom, I hear a new COVID dropped." Like, all right. If the teenagers know about this, then I should probably get back to work. And this is when my phone started buzzing. And so, you know, Thanksgiving weekend, I made it all the way to Friday night before I had to answer my 150 emails that I'd already gotten. And why were we freaking out? It was because of this new variant called Omicron. So why were we so upset about Omicron? I mean, we had already had Alpha and Delta and all these things, but Omicron was even more mutated. So here's what I showed you a minute ago, all the amino acid changes, 10 or 12 changes in spike for all these other variants. Well, Omicron had 30 changes. And not only that, but it had a bunch of the greatest hits, or maybe you would call them the worst offenders from what we knew from before. There were mutations that we already knew in the context of the other variants made it more infectious. And there were several of our worst offenders in terms of immune evasion. So we were really worried about this. And so we and others just worked around the clock in December to try to figure out what was going on. And here's what we learned. So what I'm showing here, this is actually not from a clinical trial. Now, these are actually volunteers who are NIH employees who had just gotten vaccinated because we were eligible to get vaccines, right? And so here's what their neutralizing antibodies look like against D614G. That's our original strain, right? And everybody has antibodies over around 1,000, which is good. We know that that's protective against the original strain. If we compared that to beta, it is about 9 times less. So still neutralized, but at 9 times less. But then when we looked at Omicron, on average, it was 35 times less than the original, and about half of the subjects didn't recognize Omicron at all. So this is exactly what we had feared when we saw the sequences. But the silver lining is that when we looked at people who got that third dose, people who had gotten their booster shot, things actually didn't look so bad. I mean, we still saw a drop. So beta now is only 3 times less, and 6.5-fold less for Omicron. And if you look at the actual numbers, this is now almost 1,000, right? Similar to what we saw back here. And this is the amount that we thought that we knew was protective, at least against the original virus. So it looked like giving a booster shot was really, really the way to go. So we were going to see some loss, but having that booster was going to be really critical. But what booster are we using, right? So currently, even today, all the booster shots that you can get, I'm actually going tonight to Safeway to get another booster shot, was to boosting with a different vaccine or the same vaccine. So if you got J&J, you're being told now you should get boosted with Pfizer. And if you got Pfizer first, you can get boosted with Pfizer or Moderna. You can mix and match, and it's just fine. We've done studies showing that mixing and matching the different vaccine platforms, the different companies, that's just fine. But everything uses that original spike sequence. It's the sequence from Wuhan from the end of 2019 with 2P in it. It's all the original spike sequence. But should we be boosting with the variant sequence? Should we be boosting with an Omicron virus? And actually, Moderna's trying that right now. But we've also tried it with beta back last year when we thought beta was the really bad one. So how do you test this? Well, you boost people with the original versus you boost some people with the original and you compare them to people who you boost with the variant or with a mix of the original and the variant. And so in people, you're just going to measure antibodies because it takes months and months and months and months and millions of dollars to test it in 30,000 people and look for efficacy, but you can look at the antibodies. In animal models, you can do it kind of faster. So you can measure the antibodies. You can also look at protection from infection because you can infect an animal when you want to. You can control that. So first I'll show you some of the data from the animal model. So these were non-human primates, the rhesus macaques, that were vaccinated with the original Moderna vaccine, 2 doses. And then we let them sit for like 6 months, just waiting around for, let's see what the next variant is, and then we'll deal with them then. And so the next variant in this case was Omicron. And so they were boosted either with the original or with an Omicron version of the original that we also got from Moderna, or a control. They weren't vaccinated at all. And so you infect the animal, you vaccinate the animals, you infect them with Omicron, and then see what happens. And so you measure the amount of RNA that's in their lungs. And so the animals that didn't get vaccinated had lots and lots of virus in their lungs that they slowly clear out, and they got sick. But the animals that were vaccinated had very little in their lungs. It was cleared very quickly, and they didn't get sick. And everything looked the same, whether we boosted them with the original, shown in red, or with an Omicron vaccine, shown in blue. And I don't have time to show the data, but the antibodies also look very similar. The antibodies in their blood also look similar. And in a previous study, where the same exact experiment was done, but using beta, same story. Either the original or the variant worked equally well in the animals. Well, animals are fine, but what about people? And so we looked at people who were boosted with the original or a mix of the original plus beta, right? Because there's always a little bit of a time lag. And so we tested when they were boosted. Here's their antibodies against the original and their antibodies against Omicron, shown in red, if they got the original vaccine. And if they got the original plus beta, it pretty much looked the same. So similar to what we saw in animals. There was a follow-up study with more people that is up on a preprint server, showing that actually the beta worked a little bit better even than the Omicron, but not huge, not like 10 times better. And so the implication of that is for our current vaccines, it's probably not worth the effort to change to a variant vaccine for boosting yet. For people who have never been vaccinated, that's a whole nother story that I won't get into. For them, that you probably do want to switch it. But one of the problems is with messenger RNA, you can change it pretty fast. You can change things and get stuff out within 3 months. But the viruses are changing every 6 months. So it's a little unclear how that's all going to go. And so things are changing fast. So here's that same graphic again, showing the sequences around the world. And so originally there were the ancestral forms, and then alpha, and then delta. And now here's our Omicron forms. And there's multiple forms of Omicron. There are different Omicron sublineages. So the original one was called BA1. Then there was BA1.1. Then there's BA.2. And another one, BA.2.12, that is homegrown. It started in New York. Isn't that great? And so far, we're okay. So BA2 is not actually worse than BA1. They're pretty similar in terms of their neutralization. This is looking again at people who were vaccinated with 2 doses or 3 doses. And if you look at their neutralization capacity against BA1 and BA2, it's pretty similar. So that's okay. That's a little unpublished data of ours. What's next? So, well, what's probably next is something called BA4 and BA5. So here's globally in the past 6 months, BA1, BA1.1, BA2, BA2.12.1, which is what's dominating New York right now. In South Africa, things look different. In South Africa now, we've got a wave of something called BA4 and something called BA5. And BA4 and BA5, we're seeing them in the US. We're seeing it all over the world. It hasn't taken over yet. And so we don't know if it's going to be like beta and kind of stay in Africa, or if it's going to sweep the world. And so we're watching this very closely. So we'll know pretty soon whether we need to be worried about these. And so we're starting to get data, very preliminary, on how bad these are. So this leads me to some of our open questions. So, what are the correlates of protection for variants? We know pretty much, how much antibody you need to protect against the original virus, but we're not sure how much antibody you need to protect against these variants, and that's something we're working on now. Do we need to keep giving booster shots? Is this going to be like flu where you have to give it every year? Is it going to be worse than flu where you have to give it every 6 months? That's probably not a sustainable model for a global pandemic. So can we make a vaccine that's better, that we don't have to keep boosting? What's the next variant going to be? And will the current vaccines continue to protect us against the next variants when they come? And then on a more molecular biology level, which is some work that my group is doing, is why does a third dose improve breath more than cross-reactivity more than a second dose? Why was that booster dose so important? And so we're doing some detailed B cell lineage and genetic work, looking at affinity maturation of B cell lineages over time. So to summarize what I've told you today, previous coronavirus studies and mRNA vaccine technology in younger and older adults, and again, this is very important for older adults, vaccines containing the original sequence do elicit antibodies that recognize the variants, but at lower titers. And boosting with either the original or variant sequences improves the responses to variants. And so here's my public service announcements. Vaccines still work. They do still protect from severe disease. The current vaccines do have lower efficacy against the Omicron variants. And you can tell that because we're back up to 100,000 cases per day. And cases are raging in New York City again, even though a lot of people are vaccinated. That doesn't mean that the vaccines don't work. The vaccine still cuts the risk of infection in half. And half of a really huge number is still a really big number, and that's why there's still so many cases, but vaccination does still cut your risk of infection in half. But most importantly, it cuts your risk of hospitalization and death in half, so, or even more than that. So even 2 doses of the vaccine, so not even the booster, but even 2 doses of the vaccine gives you just 1/3 the risk of being unvaccinated for hospitalization or death. And then I found this on the CDC website yesterday. Boosting is even better. So adults who received 3 doses of a COVID vaccine were 94% less likely to be put on a ventilator or die from COVID-19 compared with adults who were not vaccinated. So here is my advice to you. If you have never been vaccinated, please do so now. If you are vaccinated but haven't gotten your 3rd dose, get that dose now. It's going to help you against Omicron a lot better than just the 2 that you have. And if you are over 65 or over 50 with some sort of risk factor, like you have COPD or diabetes, go ahead and get that 4th dose. So with that, I'd like to thank the many people who have worked on this. My team, particularly C.G. Odell and Steve Schmidt, my colleagues at the Vaccine Research Center, Kazmika Corbett, Barney Graham, our leader, John Mascola, Ling-Shu Wang, and then everybody who's on this list. My collaborators in the US and internationally, particularly my neutralization assay support group, particularly David Montefiore and Penny Moore and Lynn Morris and Janelle Beeman in South Africa, and my pandemic office mates. And I'll stop here and take questions. Thank you.
Wow. Thank you for that talk, Nicole. I'm just amazed. At the same time, I'm happy that I personally have the fourth booster. So let's see if there are any questions in the chat room. I've asked people to ask questions there. I don't see any questions coming in, but let me start off though. The COVID-19 was preceded by SARS and MERS. Do these are those sequences similar enough that these vaccines would also protect against those diseases?
They are similar, but they're not similar enough.
Not so similar enough.
They're similar in that you sometimes get antibodies that are cross-reactive. So you can get antibodies from people who. So we actually looked at people who were survivors of SARS to see if they had antibodies that could neutralize SARS-2.
Yes.
And a few of them did, just a few, and we were able to isolate monoclonal antibodies actually that could cross between the 2 viruses, but they're pretty rare. There is a lot of work to try to make what we're calling a pan-coronavirus vaccine, a vaccine that could protect against SARS-2 and SARS-1 and MERS and all these related viruses. That is a major goal of many researchers right now.
The massive structural change in the spike protein on infection is incredible. Do you know of any precedents for that large change other than, say, allosteric proteins, perhaps? Was that unexpected or is that expected?
This is expected. Virus spike proteins tend to do this. So the HIV protein, that's my day job, what I usually work on is the HIV envelope protein.
Right.
That undergoes several steps of a very large conformational change similar to this. The inspiration for Jason McClellan's work on this was the respiratory syncytial virus. This is a virus that causes pneumonia, especially in premature babies. And so he had done some previous work showing that that you had this very large conformational change, and that it was critical for making a good vaccine to have that prefusion form. So in virology, this was already established. In influenza was the first example of this, actually.
Right. Okay. So if you did an NMR structure of one of these proteins, would you see any of that movement, or would it be frozen until they actually bind?
Well, I've seen animations of it. I don't know that it's actually been captured. NMR is a good technology for looking at things in motion, but I think these proteins are too big, actually,
Right.
to be captured by that particular
It's too large.
technology.
Yeah, understand. So some questions have been coming in. So let me go to them. Is there current research showing efficacy of the fourth dose and the variants that are being watched in Africa and elsewhere?
So most of the data actually comes out of Israel, where lots and lots of people are getting the fourth dose, and you do see improved efficacy. So whereas people with 3 doses, the rates of getting hospitalized was starting to come back up. When they got the fourth dose, the rates came back down.
Right.
So that's against the current viruses, which is Omicron BA.1 in Israel right now.
Okay, so the next question is there a molecular biological reason why the variant vaccines, I'm trying to parse the question, do not have efficacy compared to the initial vaccine? I'm not sure that I don't understand the question, because the vaccines are all the same.
Right. So we don't have. So the variant vaccines, we don't have human efficacy data for them yet.
Yeah.
There are trials being done in a few dozen people right now. In terms of the animal models, the efficacy is the same. And in terms of the antibodies that you get, which is a stand-in for efficacy, they do look the same, actually. So I'm sorry if I was not clear about that in the talk.
So the last question in the chat room, at least, is if this is going to be like the flu vaccine, where every year it's a new vaccine, what would be the strategy? Mixed variants, or sequencing of every variant continuously done? What's the current wave in New York? What is it
Okay. So the last thing first. So the current wave in New York is BA.2.12.1. I'm not in charge of naming these things. But yeah, so it's BA.2.12.1. That's the first question. Let me back up. So sequencing of every variant is continuously done. Some countries have better sequencing than others. We've been very lucky to work with some really phenomenal scientists in South Africa who are monitoring what's going on in South Africa and Botswana and other nearby countries. But their capacity is not great. They can't do tens of thousands a day. The best sequencing actually comes out of the UK and Denmark. And then we've built over the past year, we've improved, we were kind of flat-footed in the US, and we've been building our sequencing capacity. And so, yes, the sequencing is done all the time. Any person at, any NIH employee who gets infected gets their sequencing done, kind of thing. Right. And so back to the probably the most important question for people's lives is if there's going to be like the flu vaccine, it might be a new vaccine every year. Is it going to be a different one every year, or is it going to be mixed variants? And I think the answer is it's going to be a mix. It'll be a mix of like original plus that year's variant, the most recent variant that we've got. And the reason for that is because we don't know what the next variant is going to look like. And it might be that the next variant is different from the original, but has changed in a different way than Omicron has, right? So the next thing might not look like Omicron. It might look totally different, go off in a, if Omicron's over, if original's over here and Omicron's over here, it could be off in a totally different direction, right? Instead of going east to west, it's going north, right? So you're going to want to have something that's central. If we can do this geographic analogy here, you'll want to continue having, in my opinion, you want to continue having that original sequence in there, because we know that works kind of okay for the variants. And then mix that with your newest variant so that you have the best protection against that.
Good. So the last question that I see here is, how about varicella mixed with other ones you mentioned? I don't know what varicella is, but you may.
Yeah, actually, I'm not familiar with varicella, so
Okay,
I'm sorry.
So Anjana, you'll have to
Anjana, you'll have to
clarify
tell me.
that if you
Yes.
go on. Is there an understanding of why COVID vaccines seem to be less protective than vaccines for viruses like polio that seem to stop the virus totally, besides
Right.
the rate of variant mutation?
So hi, Andy, and that's a great question. So if you look at respiratory viruses generally, you generally don't get protected for life. You get reinfected all the time. If you look at other so there are 4 common cold viruses that are actually coronaviruses in people. 2 that are pretty close to SARS-CoV-2 and 2 that are in another group. But when you look at these coronaviruses, you get reinfected again and again over your lifetime. So your immune system does not naturally give you lifelong immunity, whereas polio, you do. So a lot of the most successful vaccines, the vaccines where you do get lifelong immunity, it's where an infection gives you lifelong immunity, and you don't have that for respiratory diseases. I've also seen a hypothesis that I'm not sure about that has to do with whether the virus is confined to being respiratory versus being systemic, being in the other organs of the body. That's not clear. So that's what we know. That's what we know. It's still, if we can figure out how to get lifelong immunity for a respiratory virus, that would be ideal. And then we wouldn't have to get these boosters. We wouldn't have to get a flu shot every year. And that's definitely a goal of a lot of research groups, but we're not there yet.
How strong is the evidence of efficacy for the booster shot for children?
Actually, I have it in my list of things that I need to read. That data just came out. So the booster shot for kids. So the booster shot for children just got approved yesterday, or got recommended by it got authorized by the FDA and recommended by the CDC just yesterday. And they actually waited. They waited to get some additional data on what the booster looked like. In terms of antibodies, if antibodies are stand-in for efficacy, in terms of antibodies, the booster looks much, much better. And actually, kids respond better to the vaccine than adults do. Kids respond just great to the vaccine. But it looks like in terms of antibodies, that just like in adults, in kids, having the booster really makes a difference in terms of being able to recognize the Omicron variant. So that's the evidence. In terms of efficacy, getting really sick is so rare in kids, luckily, that it's hard to do a study with enough people that you actually accumulate enough really bad cases to actually be able to tell the difference. So I think the efficacy data is pretty soft, but the antibody data is really clear that it's worth it.
Okay. Next question is, data show a decreasing antibody levels over time. Can you speak to any research being done on memory T and B cell development in the response to vaccination or infection?
Yeah, there's a lot of work being done at the molecular level. So a lot of looking at what happens to the frequency of B and T, virus-specific B and T cells over time, and also how are antibodies maturing. There's evidence that you get a lot more maturation after a 3rd dose than after a 2nd dose. We're also seeing some differences in people who got vaccinated versus vaccinated but had an infection in the middle. I've got an ongoing study on that right now, and hopefully in a couple of months I'll be able to tell you in more detail what the answer is.
Right.
But that's definitely an active. There's actually a very active area of investigation.
Okay. Next question is, Omicron is more infectious but less effective in causing severe disease. I didn't know that, but do you expect more
so
viral
that's actually not true. So this is actually a
true.
misconception. So early on, it looked like Omicron was less severe, and there's two pieces of data that said that it was less severe. One is that it's less severe in all of our animal models. Like it doesn't, hipsters don't get sick from it, and the monkeys they don't get as sick. But in people, Omicron was first detected in South Africa, and the cases were more mild. But most people in South Africa had already been infected in the previous waves. People had already had Delta or Beta. And so it was unclear whether that apparent mildness was because people already had some pre-existing immunity. And same thing in the US, 75% of people are vaccinated, or at least have gotten started to be vaccinated. And so that's going to reduce it. But if you look carefully, just at people who have never had it before and never been vaccinated, it's actually just as severe. It's actually just as bad, which is why hospitalization rates are going back up, especially in areas where vaccine rates are low. But then you have a philosophical question, and this is actually a very interesting question. So the question in the chat is, do you expect more viral evolution is in fact beneficial to the virus? More philosophically, why virus will mutate more, or maybe a different gene might suddenly start mutating? So there's two kinds of pressure, evolutionary pressure on the virus. One is adapting to the human host, right? This previously infected bats, right? And so it has to change in order to efficiently spread from person to person. And so those things are going to happen continuously. And every time we see something that's like so contagious, we think, oh, this is as contagious as it's going to get. Delta's as bad as it's going to get. It's not going to get any more contagious. And then Omicron did, and Omicron is more contagious. So it's definitely in the virus's best interest, if you will, to become better adapted to spreading from person to person, not necessarily being sick. And in fact, it can be advantageous to the virus to be asymptomatic, right? If people are spreading the virus and don't know they're sick, people are just going to go out and go to parties and never know they're sick. So that, in that way, that can be, being less virulent can be advantageous. On the other hand, some people are what you call super spreaders. Some people are really, really efficient. And if maybe they're coughing, right? So something that's going to make somebody cough a lot is going to make them spread the virus more. So there's sort of opposing forces in this, which is one of the reasons it's been so hard to predict what the virus is going to do. I mean, the only thing we know is that it's going to keep mutating, but to predict how it's going to keep mutating has been rather difficult to predict.
So with that last sentence, that it's going to keep on mutating, which we assume means you're going to continue to have a lot of work to do, and your colleagues.
Yes. Yes, we can't all go back to our day jobs yet. So a lot of researchers, we pivoted from working on the viruses we were already working on to working on COVID. Right. And it's been, so especially people in HIV and people in influenza have started working on coronaviruses. And in some ways it's exhausting and we'd love to go back to our day jobs. On the other hand, it's been really nice to have a vaccine that worked. Yes. So I'm going to leave it. I'm going to leave it with that we have a vaccine that works. And yes. So my advice to everyone is get vaccinated. Yes,
it has to feel really wonderful to be part of a process that has saved millions of people's lives. And that's not an understatement after all. Yeah. Before we close, I'd like to thank our sponsors who helped get an audience for this: the Bell Zeller Scholarship Trust Fund, the Biochemistry Doctoral Program at the CUNY Graduate Center, chemistry departments in Hunter and Lehman colleges, the Hunter College High School, the CUNY Academy for the Humanities and Sciences, CUNY Graduate School of Public Health and Health Policy, and the School of Health Administration at Dalhousie University in Nova Scotia, Canada. So thank you to all of those, but most of all, thank you, Nicole, for your wonderful talk. It's been my pleasure to be here. Thank you. As I indicated, this has been recorded. We do intend to post this elsewhere, as a questioner just asked. This will be posted, and we'll tell the people who registered about this posting, presumably on YouTube. That's what we've done in the past. So again, thank you so much. It's been a wonderful talk. Right. Okay. Thank you. Thanks everyone for coming. Okay. Thank you so much. Okay. Bye-bye to all.